yesterday. Ooh.
Okay.
Might be live.
I'm still here. Okay.
But I said yesterday that we might want to mute our microphone when we are not speaking, so that we make it easier. And like if there's a lag, it's all right.
Okay. So they are saying we are live. I'm a little bit confused. I don't know if the chat we're seeing is everyone's chat or just us, but I guess if we're live, we're live. Is for the organizers, is there a way to share slides? I don't quite see it through this interface. Oh, there I see it. It's share screen. Ooh, it's share the entire screen. Give me a second before I do that. Way too much stuff open to share on the screen. Excellent. So, welcome everyone. Let me see if I can get the slides up on a single screen for sharing. No, I don't like that. Oops, no. That means I can't see anything. How did I just do it? Sorry. Okay, this is probably the easiest right now to let me actually. Let me hope I'm sharing the right one. Oh, this is making it hard. So, it seems like I'm having fun security issues on sharing the screen. I have to quit Firefox and rejoin, so I am not doing that. Does anyone, I actually shared the host link to the slides to everyone. So if there's any way for someone else to share the screen, that would be lovely.
I'm gonna try.
Meantime, yes. In the meantime, as the theme of HBM is this year, everything works. It just takes a little bit of extra time. So welcome everyone. As a bit of background, we have been running sort of these multi-echo fMRI gatherings at in the Open Science Room for a bunch of years. I'm trying to think of it's like 4 or 5 years now. And it kind of started out as just this small group of people saying, okay, there's other people doing multi-echo fMRI. What do you think of it? And just getting to know each other. And it's grown to be sort of a chance to sort of have discussions and get it, help other people learn a bit more about multi-echo fMRI and kind of build a community. So in that theme, this year, I think one of the big changes in the last year has been multi-echo fMRI is still sort of a small fraction of the field, but I've been seeing more and more people just as users, that there's a growing community of people who are just, deciding on their parameters and they're saying, hey, multi-echo fMRI is something that I want to use. And even a few big data studies, I've had some conversations with people saying, we might actually want to use multi-echo fMRI for our big data studies. So I think that's kind of the big change this year. So I tried to bring in a bunch of people this year to talk about multi-echo fMRI in practice. So we have a few grad students who have either working on methods for multi-echo fMRI. I guess everyone here is working on methods at some point in some way, but also applications. So everyone's going to present a little bit of the slides. a little bit of slides if we ever get them open, and then we'll have a discussion. And if we can't get the slides open, we'll have a discussion without slides, which is a little bit aggravating because I know everyone is working late at night to put the slides together. So from the other speakers, does it sound like slides aren't going to happen for now? No.
I can try on my side and maybe in the meantime introduce. Oh, is someone's sharing?
Mm-hmm. Oh, someone has slides, yay.
Does that work?
I can see the slides.
Okay.
Hey, thank you, somebody. Okay. Hey.
Did I have it in screen sharing mode just a second ago?
It is in screen sharing mode, but it's we can kind of see the small slides in the center and the just sort of the small thumbnails on the left.
How about now? Is it? Let's see. Is the sound?
That is probably good enough if we don't want to fiddle anymore.
Okay, I was saying, I don't know if I can get it much better than that.
Okay.
If I put it in presenter mode, it doesn't change.
Okay, so these are all the presenters here, and we're going to go in that order. And obviously, this is recorded, so you can look at our handles and the posters that we're doing that have something with multi-echo later on. I just wanted to put them all in one place. So next slide. So I'm assuming that not everyone here knows what multi-echo fMRI is. So I'm just going to give a very brief intro. In what's more common functional MRI, which we can call single echo fMRI, you have an excitation pulse. You wait a bit of time, that's called the echo time. And then you have a readout period where you collect data, and then you keep repeating. So that excitation, wait, and readout could be per slice, per multi-slice, etc. With multi-echo, instead of waiting, we start collecting data immediately as soon as possible after the excitation pulse, and then we keep repeating. So in this example, we're doing 3 identical readouts in a row, which means instead of getting a single echo, we're getting 3 echoes after that 1 excitation pulse. And as you can see in this example, it takes a little bit more time, but there are ways to shorten it. And what that means is instead of getting 1 echo of data, you get multiple echoes. So the pictures on the right are an example of multi-echo data, where you can see the first echo tends to be a little bit more structured and a little less dropout. The second echo in this case is going to be a little bit more dropout and a little bit more bold weighting. And then the third echo is a little bit less. Now, why is this relevant? If we want to go to the next slide. So this is an example that Taylor Salo set up recently, which is really cool. So at the very top of this figure, we have, we're advancing across time. And you can see the little dot going up and down across time. Now, the signal changes across echo time. So if you have a T2 signal, T2 star signal, which is what most bold signals are, which is what bold is included in, on the left, you can see that the shift across echo is shifting the curve of how the signal changes across the echo. And on the right, you have the S0 changes, which is usually noise for fMRI. And you can see that sort of there's a baseline shift on the right. So I'm not going to get into the math of how this is used for noise removal at all right now. What I'm going to say is you have, when you get multiple echoes and you're sampling these curves at multiple points, you are able to effectively empirically distinguish T2 star from S0. And on one of the big questions of sort of, actually, I'll keep going a bit for now. Next slide. And like I said, I'm not going into more content at all right now. I just wanted to give us a brief intro. But I'm involved with the TED project, which is software, but it's also been a lot of resource building. So if you want to learn more about multi-echo fMRI, we've been putting a lot of information there. And I also put a link at the bottom to the symposium on multi-echo fMRI and applications from last year's HBM. So next slide. The charge I gave to the other presenters here, and we haven't discussed much beyond this because this is an emergent session and kind of everything is formal and casual, is why are we working and using with multi-echo fMRI? If they have any existing multi-echo work they want to highlight, and if there are any open echoes in the field that they would love to see addressed either by themselves or by other people. And with that, I have spoken enough for now, so I don't want to leave room for discussion, so I'm going to mute myself and we can go to the next slide. And I think I put Niku first. Actually, I'm embarrassed. We didn't even coordinate to pronounce names, so I really apologize.
It's okay. No, you nailed it. Don't worry. So yeah, hi everyone. I'm Niku. I'm relatively new to the multi-echo community. I'm a final year PhD student at King's College London, and I've actually, the reason I'm fairly new is because I actually work with a different pulse sequence compared to the vast majority of people, which is known as looping star. It's a silent fMRI technique that has multi-echo capability, and I'll talk a little bit about that in a second. And because of that, it's meant that learning how to use things like Tadana with a new sequence that's actually 3D and not slice-oriented. It's also radial and not Cartesian. So there's been a lot of really interesting sort of elements to that we've been exploring. So yeah, hopefully I am now heavily fixed into the multi-echo community. Yeah, I look forward to getting to know you all. So on to the next slide, please. So why am I working on multi-echo fMRI? I would hope that being part of the multi-echo fMRI community right now means that everyone agrees with this, but ultimately it's an opportunity to maximize your bold sensitivity across brain regions, explore the effect of new fMRI pulse sequences or different parameters that you want to use, and how physiology then ties in with that. I think there's a lot of really interesting scope to explore the brain with this kind of technique. Of course, understand contamination from non-bold sources, as Dan explained just now, and also boost your TSNR with echo combination. You can either do that with the accessible software that we're gonna hopefully hear more about today, or through scripting. Like, you know, simple echo combination, just add it and divide it. It should be fairly straightforward. So next slide, please. So here is my shameless plug for my poster, which is poster number 1400. We have been working with Looping Star for a few years now, about three, 4 years. We've been actively working on it. And what I'm showing at OHBM this year is that we're actually trying to not only address the high acoustic noise that comes with EPI with this technique, but also limit the impacts that having additional echoes has on your TR. So I have been working in collaboration with UCSF, and our colleagues there have developed a low-rank image reconstruction technique called Extreme MRI. And through that, we're able to sort of undersample our data to the point where we can accelerate the scan and also retain some functional sensitivity. And I think this is a really interesting sort of area that multi-echo fMRI can move into with the sort of advancing techniques of multi-band, multi-shot. Sort of how can we keep those the usefulness of those echoes whilst also getting really fast scanning. So I think that is a really interesting challenge. And if you'd like to know a bit more about the technique, feel free to come to my poster. I think I'm meant to be online tomorrow at 5 p.m. British summertime. I'm in London, so you can figure out the time zone translation. Or if not, then do feel free to contact me on Twitter or by email if you want it. I've seen a comment about the silent sequences are super exciting. Thank you very much. We actually, another shameless plug, we actually published a review paper earlier this year, and I'm more than happy to drop that in the chat if you're interested too. Next slide, please. So this was just a few of the posters that I thought were really interesting that have popped up at HBM. I mean, there are many, but I think in these cases, they sort of address, we're starting to see the areas that are starting to be addressed, and also where we maybe are going as a field. So sort of standardizing data cleaning strategies. I know Tedana is now part of fMRI prep. So there's sort of new things happening in the pipeline development to sort of clean the data and what noise actually means with multi-echo fMRI data, which I think is a really interesting avenue. The other really important thing is accessibility of multi-echo methods. So not every scanner in all of the OHBM community, not every institution has access to things like multi-band, multi-echo, multi-shot. So how can we as a community move forward when our accessibility isn't, necessarily being addressed as a sort of modality? And equally, just having awareness of the benefits as well as the applications that it can be used with. I mean, it really is a free lunch, multi-echo fMRI. You are just getting more echoes, more information within the same scan. But I think there's so much, again, personal opinion, I think with a lot of studies, people are very used to things that are well established. And I think that's a great, situation to be in, that we do have things that are well established, but are we gonna maybe move away and explore things that might even be more useful? And recent studies, on the next slide, I've got literally a whole list of references you can check out to see all the really helpful new studies coming out showing that multi-echo fMRI may be better than single echo. So check out the references. I'm more than happy to chat in the discussion section of the presentation, but I think I've said enough. And yeah, thanks for having me.
Okay, thank you. That was wonderful. I think I put Sarah next. Yes.
Yes. Perfect. So we, do a little bit more of application-based, but also kind of in methods. We're looking at multi-echo ICA, specifically just how it improves correspondence between multimodal fMRI EEG. And so a lot of what we've done is kind of preliminary work into the multi-echo ICA field, but we've started just looking at how well processing a multi-echo ICA data set compares with a conventional or a more typical pre-processing pipeline, and correlation with EEG. And we've looked at various frequency bands. For the sake of the poster that we have at the conference, we are looking at an alpha theta ratio, which is a well-known vigilance measure in EEG that can kind of tell whether somebody's like awake to drowsy sleep. And so we were able to show in a lot of these frequency bands and alpha theta that we're seeing a lot stronger correlations with multi-echo ICA, suggesting that maybe it is helping improve fMRI to more of a stronger correlation with a true neural signal. We're also seeing in predicting behavioral performance, so whether somebody might respond to a task based on a fMRI vigilance index that we have been working on, that if we use a multi-echo ICA pre-processing method, we can actually more significantly distinguish whether somebody's going to respond or miss one of these tasks. And so we have found that this multi-echo ICA has a lot of promise, and we're excited to keep kind of looking into what's to come. And the next projects we're going to be working on, kind of some of the challenges and questions that I have as a newer user for multi-echo ICA is with the parameters, default versus not default. When we're using kind of different kind of components, we see there's a lot of variability that we lose when we use the MDL component for the TED ICA, and how does that compare to the other two? And so these are kind of the questions that we're looking at addressing. When we pre-process images, we lose different degrees of freedom based on different pre-processing methods. Do we have to account for this statistically if we're comparing different pre-processing methods? And just kind of these questions are what we're starting to tackle, what we're starting to look into, and I'm really excited to kind of just hear maybe in the discussion if groups have been working on this, as some of the posters seem like they have, what their thoughts are. So excited to just kind of get to chat with a lot of people about this pipeline today. And yeah, and that's kind of my plug there for multi-echo ICA.
Thank you. I think it's Stefano.
Right. Sorry. So first of all, sorry for before. My computer got stage fright. It shut down. It never happened before. So, well, sorry about it. So While everybody else was doing methodology and pure methodology and cool stuff and nice and super cool sequences and all of that, I was playing a little bit with multi-echo in acquiring some data. And in particular, I was tasked, because let's be clear, like I ended up in multi-echo just because my supervisor said, hey, that's a good cool things that you could use to acquire some good data. And it's like, it has this and these and these feature, and it's great and it's fantastic. And man, he was right. His name is, by the way, Cesar Caballero, is in the audience. And then I saw this paper. It was from this divine entity called Prantik Kuntu, and that for me was unexisting up until today, but it gave us the knowledge of multi-echo ICA. So that's what I played with a little bit more. So what I did at a certain point in my PhD was just to ask 7 friends that are not so friends anymore to get into the scanner for 10 times over the course of 2 or 3 months, and do this simple task simple task for me that was basically just breathing a little bit, stop breathing for a while, and then breathing again. And almost none of them died. But after not receiving any complaint for the task itself, we managed to get a nice data set in which we could compare how we can measure cerebrovascular activity. That, for those of you that are not familiar with physiological stuff, it's basically the way that your brain responds to particular strength, like CO2, like excessive CO2 in your vascular system, or other things like that, general changes in gases, and basically how the brain managed to dilate or constrict the vessels in order to be able to provide oxygen to all of the neurons that we have. And this is what the task was about, basically seeing how these people could react to it and seeing how this thing changed over the course of time. And if you can go to the next slide, please. We kind of managed to do it. I'm sorry that you cannot see it very clearly, but one thing that I managed to do was basically measure this cerebrovascular activity and the lag with which it happens, because it's not like, as everything in the brain, it has like a little bit of time between what you measure and what you actually see and when it actually happens. So we measure these 2 parameters, which are CVR on your left and lag on your right. And what we saw was trying to get good maps for all of the sessions, all of the subjects. And if you can see it, very little, but maybe you can see it, you might find a little bit of difference between the maps that you see in the first row that are called SC MPR, that's the single echo, that's our second echo of the sequence, versus the OC MPR, that is basically the optimal combination of 5 echoes that we recorded. And you should be seeing that optimal combination helped us getting more straight maps, maps that had more significant voxels in general, maps that were more coherent regionally, and especially didn't have any fuzzy areas in which maybe you got an incredible contrast between positive and negative polarity that wasn't expecting, as you might get from single echo. And in the meantime, since I had 10 scans of this CVR mapping, I managed to compute the interclass correlation coefficient. That is basically a way to say how stable are things in time. And again, it's not super noticeable from the slides, and I'm very sorry about that, but trust me when I say that single echo was slightly worse, if not, well, a bit more than slightly worse, actually. Like, it was somehow worse than optimal combination, both in the CVR itself and in the lag, in managing to see the lag. So that means that optimal combination and multi-echo gives us more stable measurements over time. And finally, I also managed to do a little bit of a contrast. The last line is a linear mixed effect model. You can see it if you're not familiar with it as a comparison, as a very fancy T contrast, T stat for contrast. And what you can see there is that there is a real difference between single echo maps and optimally combined maps across most of the gray matter, but especially in the cerebellum and in the subcortical areas, and that's some areas that normally gets a bit less considered when you would get single echo. In the same time, and if you can go to the next slide, please. So these results have been published in a paper that you can check out. And actually, I went on in the paper rambling about ICA and how to apply ICA, and you can read all of it. But at the same time, another group independently did reach very similar conclusions, and that's this paper here from Alexander Cohen in 2021. He has a poster at OHBM as well, and he saw more or less the same thing, that basically multi-echo had much better TSNR than single echo. It had much better reliability over time than single echo, and especially that there were some differences between the two maps, more or less in the same areas that we saw with my dataset. So this is what I've been doing with multi-echo in practice. That has a lot of clinical applications, that can have a lot of other research applications, and there's plenty of things that I guess we want to see and we want to implement from now on. And I guess we can discuss about them in the upcoming half an hour.
And now the divine entity Prantik Kundu. I will say we're joking, but as a bit of background, multi-echo fMRI has possibly around longer than some of the presenters here, but it was kind of a curiosity. And then right around 2012, Prantik came out with a new method to use multi-echo denoising, which some of us are still sort of using the descendants of. But in general, in addition to coming out with that one method, it sparked a second look at multi-echo fMRI as a tool. And a lot of the stuff we're doing is sort of because that second look was sparked, the vendors got interested in sort of adding this, people made the pulse sequences, and it helped build a lot of the community. So, and since then he's abandoned some of this to work for this no-name company called Hyperfine, which is slowly becoming quite a big thing. So I assume I should say you're not presenting as representative of your company here, but you can say that as well. So welcome, Prantik.
Hey, Dan. Thank you, everybody, for building on this technique and finding applications. I've been out of the game for about 3 years now, but I've been keeping track, and it's just phenomenal what the community has become, and so many contributions from so many people. Yeah, I'm really looking forward to our chat in a bit. I wanted to plug a few studies that have caught my attention that I kind of talk about just briefly. I can't do justice to the papers, but I just want to kind of reflect on them a little bit. So in this slide, I have kind of addressing the concept of precision fMRI, which seems to be emerging, if not, established already. And basically what I have is side-by-side comparison of a couple of papers. One that was by Mike Lombardo, his group, and myself, and 2016. And last year, Charlie Lynch and Connor Liston from Cornell did some really amazing work. And kind of both of these papers have in common the kind of motivation that if you reduce noise, you should be able to increase statistical power at the group level. And so why is that relevant? So in this 2016 paper with Mike Lombardo, this is a response to the Button et al. Criticism of neuroscience, where you need big N for anything, any significant measurement. I mean, I'm paraphrasing, obviously. And our response was that's just not feasible for a lot of clinical studies, right? When I was working at Sinai, I did a lot of work with neuropsychiatry, and the populations that are being studied, in personality disorders or affective disorders, you can't get, in a 2-year period, you can't get more than 30 subjects, 30 patients. So, for a lot of clinically relevant studies, you have to make small subjects, small N studies feasible. And so, kind of with that inspiration, our argument was okay, we're reducing the variance due to noise in fMRI data by 60, 70% with some reliability. How does this affect sample size required to detect significant activation across regions, across different tasks. So this is in the context of autism, but this study was done in normal volunteers. Kind of long story short, some of you probably have seen this paper, but in two types of tasks, one's called the stories task, the other is called the self-other task. We also conducted resting state in this study as well. Basically, we found that reduction of non-bold noise led to halving the number of subjects required for 80% power across regions spanning all of the cortex, cerebellum, and subcortex. And so it was a surprising result where, where in some cases where you'd need, let's say, 50 subjects, in some regions where you need 50 subjects to get a significant, around 50 subjects to get a significant effect, you can achieve that in 25 subjects. Cortical regions are included too, such as VMPFC. And more importantly, in areas where a task evokes activations that achieve 80% power at well above 75-80 data set, 75-80 subjects, that's achievable at about 50 subjects with multi-echo ICA. So from a study planning point of view, I think this has some significant implications in terms of what is feasible, what clinical studies are feasible in an fMRI approach. And so the work by Charlie Lynch and Connor Leston showed this in a different way. I think showed an effect of this nature in a different way, where they basically assessed the level of reliability or inter-trial reliability with comparing multi-echo and multi-echo ICA versus what some would consider a standard in the field, which is a fast TR single echo and ICA aroma. And somewhat shockingly, they showed that, and they basically claim that 5 minutes of multi-echo is basically as reliable, or I guess as reproducible, as 15 minutes of multi-band single echo data. And so, just to quote them, these findings establish the potential utility of multi-echo fMRI for rapid precision mapping using experimentally and clinically tractable scan times, and will facilitate longitudinal neuroimaging of clinical populations. So basically the story here is that when you affect a substantial reduction of artifact, you not only increase your ability to see single individual subject or even group level improvements in statistics, your ability to do clinical studies changes, right? It takes on your study planning is kind of significantly affected in a good way. If we can go to the next slide. Okay, so there's a fair bit of work going on in the preclinical realm with multi-echo, which is really exciting. So in 2014, myself and the group at the Sorbonne that was led by Alexandre Petit and Mathieu Santin, we did a really interesting experiment. It was basically, we conducted this over like a couple of weeks when I was in Paris. And they have an 11.7T Bruker system. And Alexandra led the rodent lab there. And so we kind of asked the question, like, let's anesthetize some rodents and see if we can resolve functional networks. And one of the really interesting findings of the study was with a single rodent, with 20 minutes of imaging under isoflurane anesthesia, we were able to resolve networks as ICA components, just like we did in humans. And the networks that we found at the rat level corresponded really well to regions that were delineated in the Paxinos atlas, which was just shocking. Actually, it was really just mind-blowing that was feasible. So a number of collaborators have kind of taken the rodent pipeline, which was kind of a simplified version of the mica.py pipeline, and kind of ran with it. The most recent that came to my attention was this paper in Nature last year, where it was called Local and Global Consequences of Reward-Evoked Striatal Dopamine Release. That's from Lee and Jasanoff. And in this study, they used an evoked response from a stimulation in the left hemisphere to trace whether striatal dopamine response corresponds to bold responses, the extent to which essentially the striatal response is completely mediated by dopamine. And so the paper is really good and it's worth a close read. And they describe that there's quite a bit of regional variability in terms of dopamine response from external stimulation and striatum. But I guess it's kind of corny, but one of the things I really liked about this study was they exploited a feature of multi-echo that I think has gone less noticed, which is multi-echo bold, because you don't do low pass filtering, can resolve very long duration responses. So in this paper, they find that the striatal dopamine release results in a 200-second response pattern. So basically, they find an elevated response over 200 seconds after the stimulation in striatum related to dopamine. And from a methodological point of view, this is simply not possible with single echo fMRI, because you'd have to clobber the data. You'd have to just either polynomial detrend or low pass or high pass filter it. And so there's a range of temporal effects, I think, that can be resolved with the multi-echo technique that are unachievable with single echo. And one of the aspects of denoising that was challenging, and kind of, we can talk about it, but a lot of the low frequency noise is actually sparse, so it can be removed as one or two components, which is pretty remarkable. That's it, and that's right. Jen Evans did the first demonstration of the super low frequency variations, and she used some really interesting task paradigms. Next slide. Yeah, sorry, I'll wrap up here. Similarly, Jean Grandjean out of Singapore did a really fantastic study where he used 200 mouse data sets and made a beautiful parcellation of mouse cortex that you can see here. This was in 2018. And there's a lot of correspondence between the ICA components that he found at the group level and what we did at the subject level, and I think the parcellation that he produces. This is fantastic. Feel free to look that up. And one more result from the study. Next slide, please. So what they also did in the study is they correlated the functional connectome from correlating time series in bold data sets to a viral tracer connectivity matrix. So basically, you're looking at like actual virally traced structural connectivity versus functional connectivity. And when you can see that the matrices are comparable, and the area under the curve for this correspondence is 0.7 or so, which, it's not 1, it's not 0.9, but that's pretty high, and the significance is less than 0.01. And it's one thing to see this in human DTI data, but it's another thing to see this in kind of virally traced wet lab work. So that's really exciting. And one more slide. Next slide, please. So another in a different category, which we can talk about, and I think it's already been alluded to, work with the Danny Pines group and Monique Ernst at NIH. We had a question early on that we see these patterns in adults. What do networks look like in children? And do the number of components vary? And so in the 2018 paper in J Neuroscience, we reported that there is quite precisely an exponential relationship between subject age and dimensionality. So, and this was observed not only at the whole subject level, but it was even more pronounced at the regional level. So if you identify the areas of the brain where you had a rank decrease in component number versus age, if you actually zoomed in on those regions, you can see like shockingly exponential like correspondence that goes region by region. If you look at the paper, there's some network analysis there that compares network number versus a number of graph theoretical metrics and shows correspondence. And a plug for Ronnie Sutton's work and Nathan Sprang, they kind of took this observation and ran with it, where we originally studied a population from age 8 to 40. They took that from age 20 to 80 to see if that effect reproduced. And it did, actually. And so in this scatter plot here, the circles are our data points that were used for reference. And the blacks are, the filled in circles are their measurements of bold dimensionality versus subject age. And they went kind of above and beyond, and they showed that this metric of bold dimensionality corresponds well to episodic memory, executive function, and semantic memory. So there's actually neurobiological, or I guess you can say behavioral correlates to this dimensionality figure. That's after controlling for age. So in general, this is, the dimensionality that is detected in bold signal, I think there's a strong argument it's actually a measure of functional complexity in the brain. So, from a statistical measure, you get a valid biological measure. Okay, so that's it for me. Thank you, everybody.
Thank you. That was an awesome summary. I think we are done with the slides now, so you can stop sharing the slides. So people are obviously welcome to add questions or comment in the chat. While that's happening, I think the one thing I want to add to what's been said so far is I'm realizing I've been interested in trying to make fMRI less noisy and more robust for about 2 decades now. And the challenge with so many of these, that's called denoising methods, is you don't have a validation. And you're trying to say, my contrast looks a little bit better, my contrast looks a little worse, or my results, or often a lot of these analysis methods are like, this looks prettier. And we have put some numbers behind what prettier is, but effectively this is prettier. And I was a little bit late to this, but I realized that one of the huge things with multi-echo is it provides an inherent and easy validation to what makes fMRI, what makes a method better. So if what you're doing is better, then you should probably end up with more T2 star weighted signal after you do it than before. And that's the concept of the multi-echo ICA method and the Terana method. It's descendant of the Terana software, but it's also really applicable to a lot of other methods. So there's a whole class of new denoising methods that, if they're applied to multi-echo data, we should actually be able to see these effects. And this is kind of one of the things I want to push forward, that if more people are collecting these methods, it should make a lot more room for comparisons and actually validating these methods and figuring out what actually works in a much more quantitative and robust way. And as a general pitch for multi-echo, it seems across a bunch of papers, if we just do a weighted average of the echoes, sometimes called optimally combined, it's going to give you about an 8 to 10% boost in your contrast to noise ratio. To get multi-echo, you might lose a few percent. So let's say you're losing 3 or 4% to do a bit better acceleration, and you're getting 8 to 10%. It's going to be best in areas where you have dropouts. So if you have areas like orbital frontal regions that you really care about, then that boost could be even bigger. Then the more advanced denoising methods, I find, do something from make things a little bit worse to no improvement to make things even like 20 or 30% better in contrast to noise. And one of my future goals is making that more robust. I would like things to never be worse. And being worse is very rare, but I would like things to never get worse, reliably get slightly better, and often get a lot better. And knowing what I know about the methods, I think that is a very achievable target. And that's kind of where I've been focusing some of my interests. With that, I am opening up for discussion. I see we have one question. This is for Sarah. Regarding the EEG fMRI study, you see also more effects in the ventricles after MICA. Can you explain that?
Yes, so that's actually something we're looking into right now. When we used, well, I guess there's two things that we're looking into. When we used compared against the conventional pipeline, we see pretty heavy still ventricle effect, and we're using the second echo, and we still want to look at kind of the third echo and the optimally combined. In terms of using multi-echo ICA and still seeing effect in the ventricle, when in regards to the vigilance aspect, we actually see that in our template, our fMRI template. And so we kind of think that there's something, whether physiologically, autonomically, something that we're still not entirely sure about, but wanting to look into as to why we're seeing that kind of positive effect in the ventricle area. So not entirely sure, but hopefully we're starting to look into that now. So to be determined, essentially. But I can elaborate if there is more to that question.
Nico put something interesting in the chat, which is worth a whole discussion itself. So do you want to talk a little bit more about that?
Sure. Sorry to drop such a huge question, but it was something like that. I noticed that last year's OHBM, when there was a whole session dedicated to variability between subjects, that actually we often attribute to noise or something that can't be the bold response, when in fact actually there's a lot of nuance in why people are different effectively. So yeah, my question, I guess, to the whole panel is just, how do you see the field moving in a direction where we can still consider that variability as something useful, but prioritize making multi-echo fMRI reliable, or maybe repeatable is the more appropriate word there. Yeah, just throwing that to the panel.
I have a thought there. I think the approach that's, I guess, in my head right now, and I'm trying to kind of figure out the best way to do this, but I think phenotyping individuals by neuropsychological measures is an important avenue. I think there might be correlates with genetic markers. I think there's a lot. There might be a lot that can be done at that kind of neurobiological level. Yeah, and I don't know how much of that's being done in terms of exploring that variability. But I don't know. That's my kind of bias in that kind of line of questioning. I'd be interested to hear other people's opinion.
If I may jump in, because that's more or less a field where I had some read about, especially in the last period. I'm not sure if I understand correctly your question, actually, because reliability is not, or repeatability is not necessarily against variability in the individual. Of course, if you consider some types of measurement like ICC or other things that we used up until now that are enforcing the fact that you should have less variability between within the individual rather than between individuals, then yeah, you're thinking, yeah, of course, like if there's variability within sessions, then definitely that's a variability that I'm losing in order to get replicability not replicability, repeatability. But otherwise, like, what I can say about it is this. There are some effects that should be global and stable, right? If you think about it, your brain shouldn't start behaving completely different from one day to the other. That's what we expect, or 1 month to the other. Then there will be changes over age and all of these things that we can consider. But there's always be a little bit of you inside your connectomics. That's what people like Emily Finn, people like Catherine Graton and all of the other that are playing with the HCP or the dense mapping are showing us with their papers, right? What we can see with multi-echo is actually seeing if using and getting better measures shows us that both that variability that we expect between individuals and within individuals, and at the same time manages to get us better and more stable measures over time. because that's what we want in the end, right? So one thing that I noticed, and like today I just presented you one subject from my collection, but like if you check all of them, like even in something that it should be so stable and so unvariable between subject, like the cerebrovascular activity, that is not a neural response. That's a physiological response. That's your vessels changing, right? That still has variations between individual, and sometimes, like, I'm sorry I didn't show the figures before, but you cannot appreciate the difference between subjects with the single echo when you can appreciate them with multi-echo. So I think that we are kind of already there where we wanted to be. And then I guess that we will have to ask Emily Finn and other people to play with our data and comes out with something that is a bit more clinical applied and more psychologically driven, probably. But
I'll add on to that. I think this is kind of what I was getting at a bit earlier, that reliability is sort of in the eye of what you want to be reliable. Like, we've known for a while that if you regress the global signal out of your data, you will really reliably get correlated and anti-correlated networks in almost everyone's brain in roughly the same areas. And they're going to look a lot cleaner than if you don't regress out global signal. So if your goal is reliability, that seems to be a pretty good step. But what we've also found out over time is that global signal also includes neural signal of interest, like vigilance and possibly other attentional and neural factors that are poorly classified but very important. So if you regress out that signal, you're essentially giving yourself reliability at the expense of some other things you care about. So I don't want to say multi-echo is the solution, but it points to one solution in this direction. So if we have a measure that says we can more accurately localize vigilance, as Sarah has shown, with multi-echo data, then we're removing a noise source that might help us get closer to the type of reliability we want for these clinical neuroscience studies. And following up on sort of what Prantik was presenting on classifying components, I do have some skepticism on, because components are such an arbitrary thing to begin with, I do have some skepticism about that idea in general. But at the same point, right now in these pipelines, we sort of have accept or reject. But we actually have a bunch of different rules that say accept versus reject. And it may actually be that why something we decide is good and why something we should decide should be rejected is actually really relevant information. So if we want to very conservatively say reject the things rejected by this rule, or the things that remain by this rule actually tell us something about individual variation that's reliable and consistent, it might give us an opening to better understand what intersubject variation is relevant, and when it varies, why is it varying? So I'm hoping that could be a potential path forward, how we could use multi-echo to better understand reliability to a certain extent.
And I guess that reliability is also incredibly important on the clinical side, right? That's probably one of the things that we should look first into if we want people to start adopting multi-echo, where I'm not going to say where it counts, but it's like a bit more where then the vendors will start actually considering putting multi-echo sequences in the machine straight away without having us to install them and going through all of the troubles to set up the sequence itself. So if I can ask you one question, because when I joined this conversation at the beginning, I was really curious to see where it was going with what are we going to do in practice to get multi-echo there. So I guess that, yeah, having replicable data, it's something great. I like having a sequence that is silent. It's incredibly good, and I would love to play with it, honestly. But I guess that we had some conversation about it already. But what else can we do to get people like our fellow clinicians, people like our colleagues that otherwise would not adopt multi-echo because they think it's too much of a hassle to go through, to actually go through it and then get their hands on multi-echo data and start using it and start applying it. I guess that the paper from Charles Lynch, the one that Prantik presented before, is actually one of the first good examples of something that we could show to our clinical peers, but and research, obviously, and research peers. But what else can we do there? Because we can do something more, I'm sure.
Oops. Just to add to that, it'd be great to hear other people's experiences in the challenges that you've had in getting the sequence going, getting the analysis going. I think that would also add to the discussion.
So we have just a few minutes left, and I'll note that Stefano's question matches nicely with the question from Josh in the chat, which is, given there are so many questions open, how do we as a community manage to answer them in an efficient way? Hopefully we can get a couple answers on that in the last few minutes of the session. I don't know if you have to shut down at 10. But if not, all of us are really enthusiastic to keep that discussion going offline as well. And there's a larger question by Cesar, and I think we will just have to answer that later. You know how to reach us, Cesar. But let's finish up on this question. So how do we, work together? Like, what do people think is the best way to go? And I'm proud that Stefano and I are kind of involved in the Tedana community. So I'd love to hear from Sarah and Nico, like, what do you feel like could have been useful? What type of community would you like to be participating in, assuming you're continuing this direction?
Yeah, it's a great point. And I think a lot of it does come down to, like I said in my presentation, awareness of what is an easy way to understand this data and what is the interpretation. That's, I think, probably the key sort of, not fear, but maybe worry about the sort of technique is we've spent so much time, we've got this whole huge literature of how to interpret single echo data. And I think really clarifying that the translation is not a huge jump between single to multi-echo in terms of how we're interpreting the data. Having the standardized pipelines there, I think is one amazing step. I think it's great that Tedana has paired up with fMRI prep, for example. That's, one route that maybe other groups and other communities can also keep in mind. But yeah, I think it's just that it's the interpretation. We don't want to focus too much on the fact that it's so different when it's not really. It's an averaging to a degree. That's one thing you can do with it. So yeah, that's my perspective. I'll hand over to Sarah.
Oh, there we go. Yeah, so I agree. I think, and I guess as a community, do you want me to speak more to like how we should be answering these questions? Is that right? Yes. Okay. So,
like what do you wish you had that you don't have from the community point of view also?
What was that? Sorry. It just kind of cut out really quick.
Also, just from the personal point of view, what do you wish you had that you don't currently have from the community point of view?
Okay.
If anything.
Yeah, I guess for me, it's just more of the ability. Honestly, it's interactions like this, places where if there's questions that I have, I could chat with somebody in a more open setting about like what kind of parameters should I use, what kind of direction should I go with this project, because we're fairly new into the multi-echo ICA realm, and so just kind of understanding one of the big kind of one thing that we're not entirely sure about is just how much variance is lost when you go and use so little components in terms of this ICA compared to a more typical denoising pipeline that doesn't that keeps majority of your components, and just questions like that other people may have or be answering that we would really benefit from just having a community of being able to maybe chat about this stuff or workshops or stuff like that if they exist. I did not previously know about it. So just kind of that things would be a great way to build community engagement. And that kind of thing is my opinion on that.
Thank you. Since we are allowed to run a little bit over, does anyone want to answer Cesar's question on what about leveraging multi-echo to improve instantaneous measures of bold signal? In other words, we always computer combine the echoes using metrics, weights, computed across the entire run. How do we move to more rapid signal changes? Can we improve the confidence of the delta R2 star, estimates in TR? There was an abstract working in that direction. I don't have any super wisdom. There's actually Stefan Hunis is working a little bit on this, on multi-echo for real time. And there was also another poster this year. I have it in the compiled list. I'm blanking on the name of the author right now, but there's another real-time multi-echo poster this year on tics and motor disorder that is trying to do some more active stuff. And methodologically, I think a lot of them are doing some variant of a weighted combination, but there's definitely room for doing much more rapidly as well. I don't know if anyone else has anything to add to that.
Just in response to the question, are you referring to this in a real-time sense, or are you referring to this in an offline post-denoising sense? Cesar?
I don't know more than Cesar's question. I'm not sure if he's going to respond.
Okay. Well, one of the things that we haven't done in multi-echo ICA yet is doing a full conversion to R2 star, sorry, a T2 star time series, delta T2 star time series. We're still in the regime of using percent signal change, which is kind of a carryover from single echo. It's one of the things I'm kind of thinking about working on, or happy to work with somebody else on, but it'd be interesting to see time courses expressed as delta T2 star, which is, by the way, not the same as one. It's not directly computed from one on delta R2 star, right? Because you need to reference the baseline R2 star.
Also, a follow-up question would be, you referred to looking at more rapid signal changes. I guess the key point there is what sort of temporal resolution are you looking for from that perspective? What is it you want to resolve? In the case of something like EEG fMRI, I completely understand because you get that full corroboration potential of the signal. But yeah, I think the important thing is to start off with what's the goal resolution you want, and then go from there. That's my thought.
So we have covered the hour. I'll also just highlight Stefan Hunis put in the chat some nice parameters. He has a nice visualization of sort of parameter space and some other cool things he's put up. So that's well worth looking at. Any last questions before we wrap up the session, either on chat or anything that people here want to discuss? Okay. Thank you so much. This has been fun. And hopefully everyone else enjoys the rest of the meeting.
Thanks, everybody.
Thanks.